By the way, the synergistic effects of heavy metals is quite complex, and this study done a long time ago, they showed that taking a non-toxic level of lead and mixing it with a non-toxic level of mercury gave a solution that had an LD of 100. So you cannot know how safe mercury is in an individual unless you know also their lead level. And I would point out that lead toxicity is very common in American children. This is a study of mercury in the saliva and feces after removal of amalgam fillings. And what this does, if you look at the data and read it in detail, is that it indicates that the major contributor to mercury body burden, pulling out in the feces, taking out with it glutathione, is from dental amalgams, not fish. Other concerns that enhance dental amalgam toxic effect: heavy metals are potent inhibitors of protein folding. As a biochemist, we talk about how proteins are made off of the ribosome and it has to be folded properly. And they're saying that the IC50, the inhibitory dose, is in the low nanomolar range. It prevents us, when we make proteins in our body, to fold them properly. That is easily reduced. This title is Low and Non-Toxic Inorganic Mercury Burdens Attenuate B-cell receptor mediated signal transduction. And the net result of this is, it says that these cells have been exposed to low and non-toxic burdens of mercury, and they've lost their B-cell receptor capability. The next one is the measurement of respiratory bursts and chemotaxis and polymorphic nuclear leukocytes from mercury-exposed workers. And at the bottom of this, they say neutrophil migration did not return to normal within a normal range, even though the mercury concentration became, quote, safe. These results suggest that safe levels of mercury exposure may lead to impairment of neutrophil function. You've already heard of this one, dental amalgam and hearing loss. This was talked about before, and I think you're getting the papers and rounds, so I'll just skip over that.
I need to ask you to wrap this up if you could. It's a very interesting presentation.
Okay, I'm just about there. Here is the problem you have with people with periodontal disease. They release hydrogen sulfide and methylthione. This is the bad, what gives you rotten egg breath. Mercury from dental amalgams will react with the hydrogen sulfide, making what's called the amalgam tattoo, or the purple gum that you will see. It also reacts with the methylthione and makes methylthiomercury and dimethylthiomercury. These are incredibly toxic compounds. We've made those in our laboratory, and we have found them to be incredibly toxic. An example of this is a tooth that had an amalgam filling and a root canal and was suffering from this toxicity. And when we took extracts of that tooth and water and put it out,
I really do need to ask you, in the interest of fairness to everyone else, I really need to ask you to wrap it up. If you want to just say your two main take-home messages.
All right, here's the last take-home message. This is the last slide, right here. What it shows here is that if you compare the brain binding, nucleotide binding profile of a normal and an AD patient, they're considerably different. If you add small amounts of mercury to the normal brain, you can convert it to have the same abnormal biochemistry with regard to the major brain protein called tubulin that holds the axon together. Mercury inhibits the same protein that's inhibited in AD brain.
Thank you for that very interesting presentation. Questions from our panelists? And remember, raise your hand if I don't see you. Dr. Griffin.
Susan Griffin, Environmental Protection Agency. Dr. Boyd, I have a question for you on your experiment in which you prepared the amalgams and then placed them in a sealed container. Could you perhaps describe that experiment a little more? You made a mold and then you pulled the amalgams out of the mold.
So we made a mold, and independent of me, Dr. Rich Fisher of the IAMT sent a box of amalgam making material to 9 different dentists. I shipped the molds to them. The molds were drilled to hold a single spill amalgam filling. And the dentist placed the amalgam in it, because we did that earlier, and I made the dental amalgams myself, and they said I wasn't a dentist. I didn't know how to do that. So we had dentists make it. They shipped them back to us, and then we would just take a plug and push the amalgam out into a test tube. And we could measure the height, weight. They were all quantified in that direction. Placed under 10 mLs of water. When we first tried to measure the amount of mercury, we couldn't because it blew our instrument up. I mean, it was off scale. So we waited until it got down, and we stopped. We didn't collect any of the real data until it leveled off. And then we did that for 28 days.
So what was placed into the test tube? What was placed into the test tube was a completely exposed amalgam on all surfaces.
Yes, a little less than 1 centimeter squared on the average, but it was a cylindrical shaped amalgam filling. And we did all kinds of playing with it. I mean, brushing it, heating it, and many things. I mean, so the amount that we did there was the amount that came off at room temperature.
Thank you.
Yes.
I have a kind of a follow-up question to that.
Just state your name.
I'm sorry, Judith Zelikoff. You placed it into water?
Yes, underwater.
Underwater. So, since we're talking about water versus saliva and the complex nature of saliva in the mouth, why would you predict that what is being released or what is being leached would be indicative of that which might be released in the presence of saliva?
Well, we weren't trying to make this a model for saliva to start with. First of all, saliva would pull off mercury a lot faster because it has a lot of hydrophobic binding sites, water doesn't. So the amount of mercury that would be collected on saliva would be even higher. The fact is, if you get the mercury in saliva, you swallow it, it gets digested, it gets in your stomach at a pH 1, the proteins unfold, they get chewed up, and that mercury would be released the same way. So saliva doesn't protect against mercury inhalation or uptake at all, I don't think.
I wasn't suggesting that saliva protects. I was suggesting that if you gave out numbers indicative of what was actually released, and if you gave it out in such a way that people might interpret that that could be the same amount, less or more, than is actually released in the mouth. If someone is trying to set up a model or give values, then you really need to set it up in a relevant situation.
Well, I think this is irrelevant. This is the bottom line. This is saying if you put it in water, which would not increase its uptake, that this would be the minimum that you would get out. If you put it into something that would absorb and bind the mercury and pull it out of its vapor form and collect it, bind it like a saliva hydrophobic molecule would, it would increase the rate of extraction.
Yes, thank you for your presentation. My question is relating to why some of the speakers have not made reference to some of the previous papers that are done in humans. For example, Berglund, Mackert, Snap, and so forth, Langworth, where they measured the daily dose of mercury release from a certain number of fillings in patients across an age range. And these were measured with mercury vapor detection instruments. Pretty well validated, and I was hoping to hear the counterpoint to the presentations, and I haven't heard these.
We did our very best. I don't know if this is on. We did our very best to avoid redundancy, and I think that Mark Richardson, who presents later, is going to address that.
But I would tell you that if you know about mercury vapor that's coming off of a tube in your oral cavity, it is mercury hates oxygen, doesn't like water, and it partitions very rapidly into the hydrophobic environments of the membranes, of the nasal, of the mucosa in the mouth, in the tongue. And so by the time you breathe it off, and by the time you stick a probe in to measure that, a good portion of that mercury would be going into the tissue. It would not be available to measure it by air. And there's no doubt about that. And I would also point out, if you looked at the data on the sheep and the monkeys, the mercury in an amalgam filling in a tooth, primarily the highest concentration in those animals was in the jawbone, or in the jaw area that they measured. That tells you that the mercury in your mouth, in an amalgam filling, is partitioned also into the jaw. So you don't get an accurate measure of how much is coming off. And you can't measure in urine. There's really very little you can do in that regard.
Yeah, it's more of a technical comment that helps me, and I don't know about the rest of the panel, but I think as we hear about the different mercury compounds, one of the important issues is to consider that comparing methylmercury to ethylmercury to inorganic mercury vapor is like comparing apples to oranges. And I don't think anybody doubts the fact that methylmercury is a toxic compound, but I think we have to put some of these issues into the right perspective. And I don't